Welcome. I'm Rich Wargo, producer for Science Horizons. I'll be your correspondent as we follow the people and the stories at the forefront of discovery, exploring where science is and where it is going. Tonight on Science Horizons, we take a look at a project which uses computation to help manage the well-being of our precious estuaries and coastal waters. And we discover amazing advances in genetic therapy aimed at finding new treatments for diseases like Alzheimer's. But first on Science Horizons, all that is alive lives by the work of proteins. And understanding how they function comes from understanding the actual shape of these molecules. Now, the Protein Databank, a single repository containing the structural information of proteins, has been created to lead science and medicine into the next century. Phil, thank you very much for being here today. In talking about the Protein Databank, I think it would be helpful if you would review exactly what proteins are, how they're made, what they're composed of, and what they do.
I think most people think of proteins as the things we eat, but in actual fact, of course, we are composed of proteins. We know now from the Human Genome Project, among other experiments, approximately how many proteins there are that we're composed of, and it's somewhere of the order of 75,000. And those 70,000 proteins have a whole range of functions: storage, signaling, structural. 30% of our protein is collagen, which essentially forms the structure of hair, skin, teeth, and so on.
Now, the Protein Databank deals with the actual structure of proteins. Can you tell us a little more about how they're put together and why it's important to understand structure?
Right. I brought some props along to help me do this. But in fact, a protein is made of 20 amino acids. So if you imagine these beads on this string, each bead, in fact, will be an amino acid. So the average protein is 300 of these long, and that constitutes the primary structure of the protein. So you can imagine if you have 20 possibilities at each position, you have 20 to the 300 different combinations in your average protein. That's actually much larger number than all atoms in the universe. So in fact, we don't actually see that. In fact, we get particular orders within these proteins, and we can actually measure those orders. If you take this string of, on average, 300, then you will have what's called secondary structures that exist along this string. So, for example, a segment of these will actually coil up into a secondary structure that we call an alpha helix. Likewise, you can have another structure which we call a beta strand. Then, of course, the real interest comes in how this whole thing folds up into a three-dimensional structure. It is through that structure that we really begin to understand biological function. So here, for example, is a structure of a protein kinase, and you can see the overall three-dimensional structure. And it's from that shape and things like the groove and so forth that actually help us really understand the function of this protein.
So that three-dimensional object you have there, the representation of kinase, that's the strand all folded and twisted and coiled into that. And if you were to string that out into just its, say, its primary sequence all the way out in one long line, how many proteins are there?
These are amino acid residues, and in fact there are 350 in this so-called catalytic core of this protein. You can also have conglomerates of these such that you have a quaternary structure in which, not in this particular kinase case, but in other proteins, where these things group together to form much more complex structures.
The Protein Databank has a pretty vast archive of structural information. How do you get that information in the first place? How do you interrogate the structure of a protein so that you can put it in the database? There
are essentially three methodologies. One is X-ray crystallography, which is where you take an actual crystal of the protein, you fire X-rays at it, you get a diffraction pattern, and from that diffraction pattern you can actually unravel the atomic positions of the atoms. Another methodology is nuclear magnetic resonance, which is a solution-based methodology in which you actually measure spectra associated with spin moments, that is how atoms are vibrating. You measure that in a magnetic field, And then you can actually, from that information, again, localize the position of the atoms. And the protein data bank contains those atomic positions. The third way is you can also model. So when you don't have any experimental data, you can actually predict what the structure would be. And there is also models in the protein data bank.
We've reviewed what proteins are, what they do. Let's talk about the subject, that the protein data bank. How did it start? What is its history?
It started about 26 years ago at Brookhaven National Lab, and it was started actually by a chemist, Walter Hamilton. And in fact, the idea was just to provide a point of collection for these different protein structures. At that time, there was only a handful, half a dozen. And it actually grew quite slowly all through, actually up until the late 80s. At that time, the technology and the techniques for getting these structures improved dramatically, and we've seen a near exponential growth rate in the number of structures. Currently, we're getting 50 per week. I mean, that was larger than the holding of the complete database in the mid-70s.
The current protein data bank as it exists today is a function of something called the RCSB, the Research Collaboratory for Structural Bioinformatics. How is that organized, and who are the participants in that group?
I think it represents a new kind of science, in a sense. I think we're seeing much more collaborative science than we saw years ago. And in fact, what we're doing here is bringing expertise from different groups. So for example, in terms of the protein data bank, the RCSB contributors are the Rutgers group from Rutgers University in New Jersey, led by Helen Berman, who in fact has developed, who is a crystallographer, determines these structures, but has also developed the nucleic acid database, which was a smaller database specializing in nucleic acid structures. So she brings that expertise. Here at UCSD, we also develop different types of databases. We also use the Supercomputer Center and the resources of the Supercomputer Center to actually compute derived information from these structures, which we then make available to the community through the databases. It's also our responsibility to distribute this data worldwide. The third partner in this is the National Institute of Standards and Technology, and their role is essentially being the permanent archive for this data so that it remains immutable forever.
Now, this is a unique archive, is that correct?
That's right. There is only one archive in the world, and crystallographers and NMR people and modelers deposit to this. We validate those entries, and then they're put in the database and they're made available on CD-ROM, World Wide Web, to anyone who's interested, in fact.
With the value that's placed on biological information and the propriety that's exercised over it so much these days, how do you maintain a flow of information to the data bank and assuring that it remains public and accessible?
Generally speaking, a researcher will submit their data to the protein data bank until publication. That is when the paper on that structure describing as much about the functions as known is actually published, then the data set itself is released. The reason this works well is because most of this, particularly the prestigious scientific journals, will not accept structural information unless there is a PDB accession code. So that means that it forces people to deposit and make this information available. Of course, there's a huge value on some of these protein structures. I mean, some are called billion-dollar molecules because their value as pharmaceuticals, and our understanding of drug design makes them extremely valuable. And you don't see those obviously appearing in the database, the ones that are done by drug companies, for example. However, drug companies are generally very good. When something loses its commercial value, they will release it and it will go into this public archive. So there's a very nice and free exchange of information amongst the worldwide scientific community with respect to this data.
As a unique archive, who are the users, or really what is the intent of the protein data bank?
Our mission, in fact, is to enable science. So that happens at all levels. And more that's important because I think biology is becoming such a multidisciplinary thing that people don't just look at their narrow area. They're now required, even though they might be experimentalists, that they need to come and look at protein structure periodically. We really want to enable that process so that they can make functional inferences from those structures in the best possible way.
Do you have any particular examples of the kinds of research that use the database?
Well, I think anyone who's researching a particular area would indeed come and look at structures if they really want to understand the mechanism at the molecular level. So if we take this kinase, for example, if I can come back to this, in fact, this is part of what's known as the signal transduction pathway. So in actual fact, you have a cell, and that cell receives some external stimuli. And that stimuli then triggers some events in the cell. It could actually involve duplication of that cell, ultimately, or it could be some toxin, for example, or some toxic chemical that the cell needed to respond to. So what would happen is that that stimuli would be received at the surface of the cell, and this particular component of the molecule sits inside, under the membrane. But what happens is it gets triggered by an event at the cell surface. That will lead it to change its shape slightly. A substrate will come and bind to it in this position, and a signal will be attached to that substrate. And then that substrate is then released, and it goes, and there's a cascade of events, and different things can happen. So in one example, you actually have a situation where a cell will proliferate. In cancer, a cell will proliferate for unknown reasons. However, what you can determine is that this particular protein is produced in large amounts during that cancerous event. So in fact, somehow this particular signaling mechanism is involved in cancer. The details of that are not known currently. But by having the structure, you can begin to get a better handle on that whole pathway.
So there might be a cancer researcher who wants to look at the structure of kinase. How do they interface with the protein data bank? How does it work?
It's actually, the simplest way is in the way that you would go to a search engine on the web. That is, you have a single text field and you would enter the phrase protein kinase. And what will be returned will be, at this point in time, 70-odd structures that relate to protein kinases, from which you can select. You can actually do comparative analysis of these kinases as well, so you could compare their three-dimensional structures to understand a little about the difference. I mean, it's really the viewing of the structure and using different depictions that really allows you to understand it better. So we, for example, use a technique called virtual reality modeling language, which is like a three-dimensional web page. So in fact, you go to the web and you can actually zoom into this molecule, and you can actually look, essentially be inside the molecule looking out. So you can actually look almost as if you can actually be the molecule, and you can actually look and see a substrate coming towards you. So that's one particular depiction. Of course, you can actually download the atomic coordinates for this particular structure, and then you could manipulate them in other programs.
Now, there's been a lot of work done with kinase. What information was discovered or revealed through this modeling and looking at the structure of kinase that we didn't know before?
The work that was done here was pioneering, and it gave the shape of the kinase for the very first time. And it turns out subsequently that other kinases that have since been, whose structures have since been determined, it turns out that the sequence, the one-dimensional homology is actually very low, yet this structure, the shape is maintained. So obviously that overall shape is very important. And what we've learned from that shape is exactly how the substrate binds and how this particular enzyme catalyzes these reactions. So we now fully understand the molecular nature of that interaction. And that's really important if, for example, you determine that kinases are being overexpressed, if too much is being produced, you can actually then design drugs that actually fit into, for example, into this groove that will prevent the substrate from binding, and in fact, allow you to then control the physiological events. So in fact, you wouldn't be able to design a drug in a concise way without this structure.
That leads right into an interesting aspect of the protein data bank, which is assisting something called rational drug design, and that's exactly what you're talking about here. Could you contrast that to the current drug design, and how does it work?
I think all the drug companies now are really pushing rational drug design because it's cost effective. That is to say, instead of taking, not at random, but from having some idea of what drug will work and testing it on animals and really looking at the effects on a complete living system is a long process and not necessarily a desirable process. So the idea of doing this on computer is very appealing. And in fact, there are now a number of examples where this has been quite successful. So for example, the influenza virus has been attacked by this process, this mechanism, rational drug design. The virus infects the cell, in fact, gets into the bronchial tubes. And in fact, the problem with viruses is that they can evolve very quickly. So you can actually have proteins in the virus that are changing rapidly. So really what you need to do is to actually get to the replicative machinery of the virus, which doesn't change so rapidly, and really try and interact and stop that process. And rational drug design has actually been quite successful in working with influenza virus.
You have a rather powerful example of how this rational drug design may be a benefit. Could you talk to that a little bit?
Well, a recent deposition in the protein data bank was a complex of what was called GP120 and CD4. And in fact, what that is it relates to AIDS. In fact, it turns out that by the year 2000, there's going to be of the order of 40 million people that are infected with HIV. That's far more than the 25 million who died from bubonic plague during the Black Death period of the 16th century. So obviously getting a handle on this disease is of critical importance. And GP120 is a protein on the viral coat of HIV. So in fact, the coat surrounds the inner RNA of the virus, and in fact, it's actually the GP120 on the coat protein that interacts with the cell, the human T4 cell, and it's that interaction that then ultimately leads to the RNA infecting the human T4 cell and ultimately a decline of the immune system. So for the first time, we know the exact atomic detail of this interaction of how the HIV interacts with the T4 cell. So this was work that was done by Wayne Hendrickson's group at Columbia, and it's a deposition in the protein data bank now that everybody can have access to and use, obviously for design studies. So obviously there's a lot of activity in trying to design drugs that potentially would prevent that initial binding to the cell.
With nearly 10,000 full protein structures, what is the size of the data bank in terms of bytes of information?
Right now, the database is about 20 gigabytes, which is 20 times 10 to, it's 20 times 1,000 megabytes. I think many people with PCs appreciate megabyte sizes. So that, in terms of data sets, particularly the kinds of data sets we have at the San Diego Supercomputer Center, is by no means large at this point. However, given these atomic coordinates, given this sort of primary data, there are many, many things that we can derive. So one of the things that we've spent quite a bit of time on, for example, is actually comparing all protein structures against each other, because we really want to understand how, classify how these structures, because it's actually a remarkable feat that nature has given us, that we have all these many, many, many combinations but in fact we actually have a relative small number of protein folds. There are different estimates on this, but there are estimates of, of the order of 1,000, so it's a very tractable number. But to actually understand why it's 1,000 requires very large amounts of computation and storage. We're also in the process of taking data directly off the synchrotrons. The synchrotron is a device that's actually used to collect a lot of the X-ray data. These are huge. There's one in Stanford, for example. These are huge football field or larger devices that are used to actually collect the X-ray data. We are in the process of getting data directly off those beam lines at that synchrotron. That's very large amounts of data. Requires very high bandwidth networking and so forth. We also have a requirement to keep these machines running for the worldwide community 24 hours a day. Since we took over the protein data bank, our access on the World Wide Web is doubling every month. We're currently getting the order of 25,000 hits a day, and that's bound to increase much further when the existing resource at Brookhaven National Lab closes down.
Can you talk about the future directions that the data bank is
Well, first of all, I think there's going to be a very large influx of data into the data bank. I think, and that's manifest in biology itself. Biology is becoming a very rapid science, and we're making great progress in the amount of information we have. So we have a lot more structures. So there is the notion of structural genomics, which is when we have in 2001 the human genome, that is this linear sequence effectively. So it's like the blueprint of life. What we don't have is essentially the three-dimensional, the buildings themselves, which of course includes us. So by actually determining all the structures that we can of the proteins that are encoded in the human genome, we'll have a huge array of information, which is actually then just another step, because then we'll have the structural basis of understanding biological function. But in fact, these things all play together. So any event that goes on within the body there are generally many proteins involved, and they are involved in concert. So the next step, so the protein data bank becomes a storehouse for this information in a large spectrum of interest. So for example, you would want to take that information and you would want to be able to mathematically model the events that those proteins are involved in, so that you can then actually be able to represent in mathematical terms exactly the events that are happening in the human body. This is way out into the future, I think. Another thing that will have ramifications in the future is the way we're trying to integrate the protein data bank with the literature. We don't want the protein data bank to be this discrete entity. We want it to be part of what I call the digital continuum. So for example, in the case of protein structure, you could actually go to an electronic version of the journal, which actually not just had a static picture of the structure, but actually came to the protein data bank, retrieved the coordinate set, and actually allowed you to manipulate the structure as you're reading the article. Well, taking that to a next step is actually having the article spoken to you by the scientific expert as you review the structure, and possibly having the structure actually synchronized with the audio soundtrack, so that you really have a good sense of the structure-function relationship as it was described by the original author in their own words. And given that, we can then take it a step backwards further, that you could, in principle, because you have the data that you've collected off the synchrotron, you yourself could actually rerun the experiment that the scientists had run to determine the structure in the first place. So I think we've got a whole spectrum of information here. There are many impediments to this, but I think in principle, this is the kind of thing that the future holds. I mean, we're very excited about this. There are technologies coming along that are facilitating this and going to make it available to everyone. What we really want to see is we want to see, K-12 and undergraduates and college students actually be able to find information about projects that are relevant to them. There's no end to where these things can go, and I think the internet is going to play such a huge role in all of this. I mean, I think we're seeing this right now through this research collaboratory. I mean, we're at 3 distinct places in the country, and yet, and you know, of course we meet at scientific meetings quite frequently, but a lot of the interaction is done either by video or conferencing or by telephone or email, of course. So this is just the beginning of what we're going to start to see. So the potential exists, if used wisely, to really put people in touch, you know, students in touch with experts, and I think just create a new realm of communication that's got to be beneficial to everyone.
If there were one thing which you could see come out of this collaboratory, one achievement, what would you dream of? What would you see happen?
What I'd really like to see is to just have a sense that this was really hitting a very broad audience, and that we were really enabling science in many different areas by providing you know, a system, a scheme by which everyone could access the data in the most useful way.
Well, this is obviously a very powerful tool in our future. Thank you very much for sharing this with us today, and it's been very interesting. Look forward to see what's coming on the horizon.
Pleasure. Thank you.
Next on Science Horizons, John Helly of the San Diego Supercomputer Center shows us the San Diego Bay Project, which was designed to help manage the health of this precious tideland.
A model is a transformation between what we observe and what we think we know about the world and our efforts to make predictions about how that world's going to behave. So the model is really the way we capture our knowledge. There are lots of things that are important to model and predict. Some of the obvious ones that come to mind are things that have either impacts on human health, for example, on the health of the ecosystem, although it's very difficult sometimes to define what that is, or that have economic impacts on the nation, the country, the world. Certainly topical in the past couple of years has been the El Nino impacts on the coastal areas. All these things affect the way in which we take advantage or are inhibited from taking advantage of the resources of the earth, or things that threaten our lives and our safety. And those are all fairly obvious ones. Scientists are concerned just in a fundamental way about trying to understand from a curiosity basis. It's part of the things that really drive science, is this natural curiosity for understanding how the things that are around us operate. You know, how is it that trees grow? How is it that, why is a tree growing here instead of over there? And what happens if we cut down you know, thousands of acres of trees here and cause erosion? What's the impact on our streams and lakes? Can we sustain a certain consumption rate of natural resources and still have a healthy natural system as well as a vibrant social system? Those are all difficult questions that are the focus of a lot of modeling efforts. Over the past few years, one of the most interesting projects that we've had has been San Diego Bay. And the reason it's particularly interesting, aside from the basic science kinds of questions that we're trying to address, is the fact that it's been driven by the need for a socio-political body to try and understand the system as well, so that they can influence, so that the data and information that we produce can influence social and public policy, and how this San Diego Bay as a common natural resource can be managed effectively or more effectively. The goal is really to understand the dynamics of the coastal zone. And San Diego Bay is a good example because it's a nicely enclosed bay, and it has all the problems of an urban setting, but all the features of an estuary. And consequently, it's a reasonable study site. And we want to take what we've learned from there and generalize it to other parts of the California coast at these fine scales of measurement, so we can start to understand better how the interaction of the watershed and the way we manage the watershed affects what happens to the coastal zone. And so we picked San Diego Bay as a test bed for this, and we combined a whole host of different kinds of data and explored different ways of mixing fish population data, eelgrass bed measurements that were made with side-scan sonar, satellite imagery to show the location of human infrastructure, with the motion of vessels, the movement of Navy ships, and their potential for interaction with the fish populations and other entities like the eelgrass, for example. And we also use the same approach then to embed the results of these models, of hydrodynamic models, of watershed models. Once we've created this virtual world, we're able to put into it processes which are part of that system under certain circumstances and look at the consequences of those events, like an oil spill. We can model an oil spill. We can model the runoff from a watershed and look at how that material is transported around in the bay. Where does it move to? How long does it persist? Where is it impinging on the eelgrass beds? Where is it impinging on the fish populations? Where do the birds spend most of their time? Those kinds of questions can then be looked at by the mayor of one city or the mayor of another city, depending upon what your point of view is. You can create a space that you can navigate. So you compute the problem one time, and then the user is able to move to their point of interest within that world. And everybody's seeing the same world, the same result, but they can view it from their particular point of interest, literally. And it's a convenient method for getting groups of people to understand a problem in common, I think. Okay, so here we have the data repository interface, and this provides access to data that we've collected about the San Diego Bay through the Bay Panel's efforts. And we'll go right to the query system, which is the method by which the user would obtain access to the data. And we can use a clickable map, which provides the main method of finding data throughout the bay. And it proceeds just by selecting a particular point on the map, in this case the South Bay, and we build up a list of locations that we're interested in having data from. We can submit that database query, and now we're going off. The computers in the next room are resolving that query, and they'll provide back to us a table showing us the kinds of data, either physical, chemical, or biological, that might be of interest in those geographic areas. Then the tables come back, and you can see that we have biological, chemical, or physical data. And we either have the raw data itself, which you can obtain through this form of a list and download it to your personal computer if you like, or you can go to these two-dimensional thematic maps, and we can see the number of hits that we've gotten in each of these categories. And if we take a look at the list of maps that are available to us, there's quite a large number, and they range from the abundance of birds on different dates. Here we have a map of eelgrass density, and we've got two color bars here. The red color bar on the left shows the density of the eelgrass, which is depicted as these red or pinkish polygons in the map here throughout the South Bay predominantly, although you can see a little bit of eelgrass up here in the north and along North Island. The greenish to blue color map is the depth of the ocean represented by this part of the map here, and then the green is actually just sea level. It represents the flat surface of the land. We don't have the elevation of the land embedded in this image. And on top of that, we've imposed a grid so we can have some geographic reference corresponding to latitude and longitude lines on this particular map. Here you can go to chemical data, which is this list here, and we have a large collection of images which we've generated from the data from the Bay Protection and Toxic Hotspots Cleanup Program, which is a state program. And we've made a series of maps showing different kinds of compounds and their locations throughout the bay. And we've also embedded in these images a chart that shows where this particular sampling date, which was 1993, August 4th, where that was relative to major rainfall events, because we're interested in also understanding whether or not these compounds may have been deposited in situ, meaning in place, or whether they were transported because of some runoff from the watershed, as we were talking about earlier. So the places which were sampled on that particular date, August 4th, are surrounded by white circles here, but the green dots that are in other places throughout the bay indicate other sampling locations, other places that were sampled on other dates. And then there's one control location which is outside the harbor in the relatively open ocean. And the color scale here shows the concentration of the compound and its location and concentration within the bay. And from this kind of information, you can get an idea of where different compounds are distributed and where they are in proximity to one another, because sometimes particular kinds of compounds will be deposited jointly. Another aspect that's kind of interesting to look at of these data are the watershed itself of the San Diego Bay. It's quite surprising to look at this and see the extent to which there's a large network of drainage systems that feed into the bay. And what we've done here is to select certain what we call subbasins within the bay and color code them so we can see their definite character and see which parts of the bay are influenced by which parts of the watershed. So what we mean by that is if it rains in this part of the watershed, the water is going to flow down here and drain into the bay at this section. If it rains over here, you can get this kind of separation in storm formation, depending upon the directions the storms come from, and how long they persist. And so the basins are really physical features of any given geographic region. And it's important to understand not only where the water flows, but how they connect, how the water network connects through a basin and finally into the receiving water of the bay. It's important to understand also the kinds of things that are being done in those parts of the watershed. It helps you understand or define the geographic area to consider when you look at the outputs of the watershed. Maybe some sections of a watershed are industrial, some of them are just purely residential, some are agricultural. The use of those areas is very much a factor in the kinds of outputs that you see from a watershed. So the big advantage of this kind of approach is that because it's public and because it's centrally located in the sense of being on a single website, people can come to one place and obtain the authoritative collection of information. And it's democratic in the sense that everybody has the same collection of information available to them to either make their own analysis, come to their own private decision-making process, and then to participate in a more public, more common decision process, all from the same base of information. And that's what's unique about this approach to sharing resource management information. And it's an approach that we hope we can extend and make greater progress with, and we're pursuing it in a number of other research areas to try and develop new and better methods for doing that. One of the most unusual aspects of the Supercomputer Center, in fact, one of the things that distinguishes this particular center, I think, is the collection of talent that's here. It's not just computers and equipment. It's really the ability to be making progress down a particular research path, running into an obstacle and be able to walk down the hall to an expert, a world expert often, in a particular area, get a question resolved that allows you to continue to make progress. It's a very difficult thing in research to be able to predict how rapidly you'll have successes or failures, or to the extent to which you'll have successes or failures. But key to being successful in a research program is the ability to get resolution of difficult problems rapidly, and the center is just fabulous in that respect for this. There's just a terrific collection of talent, and the ability to have access to that kind of talent simply by walking down the hall and asking a question is unparalleled in my experience at any of the institutions that I've worked in.
And finally on Science Horizons, Dr. Mark Tushenskiy of the University of California at San Diego discusses his work in genetic therapy with correspondent Eric von Neumann.
Dr. Tushenskiy, thanks for letting us talk with you today. Could you, in general terms, describe your area of study here at the university and what your lab is currently working on?
Okay, well, it's a pleasure to have you here. We are interested in determining how we can help promote recovery of the injured brain or spinal cord from either injury in the form of spinal cord injury, for example, or as a consequence of disorders that cause progressive degeneration in the brain, things like Alzheimer's disease. So we study a class of compounds called growth factors in the nervous system that are actually normally present in the nervous system that help promote survival of cells that are present in the brain and spinal cord during fetal development, but it turns out can be used as very effective tools for helping to prevent cell death in the adult brain or spinal cord and to promote reconnection of injured parts of the nervous system. So we study growth factors and we use genetic engineering or gene therapy as a tool for delivering these growth factors in what we call animal models of nervous system injury and degeneration. And the work that we do is, again, most highly relevant to trying to reform connections after spinal cord injury or to try and prevent cell loss, cell degeneration in the brain in what are called animal models of Alzheimer's disease in humans.
Well, why is there no regeneration of nerve tissue?
Well, growth factors are present during fetal and embryonic development. And there they don't really promote regeneration, but they promote something very similar to regeneration, which is actually helping the brain and the spinal cord establish the pattern it will have as an adult. And it turns out that to have a normal brain and spinal cord develop, you need these growth factors to tell which cells to survive and which to die, because in fact there's a lot of cell death during fetal life. And you need signals that tell which connections to go where. So these are all part of normal development, and as it turns out, they can be used after injury or in degeneration in the adult to do the same kinds of things, to help cells recover, to survive from injury, just as cells have to survive during fetal development, because their natural course is to die if they don't get growth factors during development. And some of these signals can tell which connections to go where during development, and so we can use them in the adult to tell which injured connections to go back to where they need to go to help restore function. Now that's a little bit of an oversimplification, and what happens after injury in the adult spinal cord, what happens in the brain in Alzheimer's disease isn't just a recapitulation of events that take place during development. But the sort of bottom line is that we can still use some of these important molecules, these growth factors, as strategies for helping to induce survival of cells in the brain or spinal cord after injury and to help them reestablish what were their pre-injured connections.
Well, what exactly is a growth factor?
A growth factor is a protein. It's a protein like many other proteins we have in our body throughout life. And we make these proteins, again, during fetal embryonic development, but we also make these proteins throughout life in our brain and our spinal cord. And the exact role that they serve normally during life actually isn't completely understood right now. But they're made throughout the brain in very specific parts of the brain where they have specific roles, specific functions in helping to support ongoing function and structures in the brain. For example, some growth factors are made in parts of the brain normally that are involved in memory. And if one loses growth factors in those memory parts of the brain, then memory declines. So it becomes pretty obvious that one strategy for trying to treat memory loss, to try and prevent cell degeneration in human diseases that affect memory, is to try and provide growth factors to areas that are suffering injury or degeneration. So what we do then is we take one of these growth factor proteins called nerve growth factor, and we put that growth factor into a part of the cell that degenerates in Alzheimer's disease. And we don't do this in people yet. We do this in what are called animal models of Alzheimer's disease. And we have to deliver the growth factor to the brain in some way. And these are large molecules, large proteins, and they can't reach the brain if you give them orally and they get absorbed into the circulation. So one needs some tool for delivering them specifically into the brain in the part of the brain in which they are needed. And our tool for delivering the growth factor, the natural protein, to the brain is genetic engineering, gene therapy, if you will. And what we do with gene therapy is we put the DNA that's responsible for making a growth factor into a cell, hence we genetically modify the cell, introducing the DNA that is required to make the growth factor protein. We introduce that into the cell, and then we put that cell into the brain close to the area in which we wish to deliver the growth factor. Now, the scientific term for that is ex vivo gene therapy. Ex vivo gene therapy means that you take a cell, you grow it up in the culture dish, you genetically modify it to make a substance of interest, in our case a growth factor protein, and you document in cell culture conditions outside of the animal, outside of the patient, that the cells have been genetically modified to make what you want them to make. You can verify that in the tissue dish before you ever go back to the animal or the person. Having established that, then you amplify the number of such cells that have been genetically modified, And then you harvest these cells and you transplant them back into the brain or the spinal cord to stimulate cell survival or the reconnection of injured areas of the nervous system.
When the growth factor is introduced into the patient or the host, is it triggering the cells that are already there to start producing growth factor also, or is it only those cells that you're introduced are going to be doing the healing?
Mostly the cells that you introduce will deliver the growth factor, kind of like a little pump inside the brain or the spinal cord, to stimulate the growth of connections that have been injured, or to help the cell survive some condition of stress, like Alzheimer's disease. Now, it turns out that once in a while, when you do this, when you put the growth factor in the brain using gene therapy, you actually increase growth factor production by the surrounding brain next to the transplant of the tissue. But in fact, most of the effect from this gene therapy approach is derived from putting in the cell itself that makes the growth factor. And that growth factor that is secreted from the cell exerts the effect on the brain. And as long as we've brought up the point that this is then a process in which we transplant cells into the brain, perhaps I should make the distinction that this isn't like fetal grafting, fetal transplantation, for example. In this case, we have the capability of taking cells, say, from my skin, just from a simple skin biopsy performed with a local anesthetic, growing up my cells, my skin cells, in the culture dish, genetically engineering them, genetically modifying them to make a growth factor and to make a human growth factor, then taking my same cells and putting them back into my body. So we're not using fetal cells, so there's no real ethical controversy about the source of the cells. And since they're my cells, they won't be immunologically rejected. We don't have to worry about transplant rejection. So it's a source of cells that make a growth factor that's stable, that is available anytime because my skin is always around, and cells that won't be rejected. So it's sort of a practical approach for delivering these very important therapeutic substances to the brain or spinal cord. And this work is sort of, and not just the work, of course, of our lab, but of hundreds of labs throughout the country, throughout the world, that this work in the last decade has dramatically altered the sense in which we perceive our ability to actually promote regeneration of the brain and spinal cord from neurological disease. 10 years ago, the idea of preventing cell death in the brain or stimulating regeneration was thought to be a fantasy. We thought that you're born with a certain number of cells, that in fact you start losing cells at the rate of 10,000 a day after the age of 20, and that there's nothing that can be done to prevent those processes. And in fact, we now know that's not the case at all. There is some loss of cells with aging, but we also have a number of tools that we can now use to intervene to try and prevent that loss. And we now believe that one can indeed try and promote regeneration of the central nervous system of the brain and spinal cord, and that techniques like gene therapy may be a means of accomplishing this.
What sort of results have you seen in mimicking Alzheimer's disease and spinal cord injury in animals?
Well, the idea of gene therapy for delivering growth factors in animal models of Alzheimer's disease would be for the growth factors to prevent cell loss in the brain. So what we have done in our animal studies is taken an animal's own cells, once again, we've genetically modified them to make a substance called nerve growth factor and put in those nerve growth factor producing cells in the brain in a part of the animal's brain that is the same region of the brain that degenerates in people with Alzheimer's disease. So we know which cell populations in human Alzheimer's disease are vulnerable to degeneration and death. That occurs in very specific brain regions. And knowing which regions they are, we can induce the loss of those cells in animals to sort of mimic what happens in Alzheimer's disease. So we do that. We put a lesion, we make an injury in the brain of the animal to induce the loss of certain cells. Then we transplant in the animal's own cells that have been genetically modified to make a growth factor. And we find that in animals that receive the growth factor secreting cells, we can prevent the degeneration of those cells in the brain. So this is an approach to the treatment of neurological disease that aims not to compensate for cells that have already been lost, but instead aims to prevent the loss of those cells once a diagnosis has been established. Now, Alzheimer's disease is the most common neurodegenerative disorder. It affects 4 million people in the United States today, and that number is predicted to double in the next 20 to 25 years as the proportion of elderly people in the U.S. population increases. And there's really not an effective therapy for Alzheimer's disease that aims to prevent the ongoing cell degeneration that occurs in this disease. And it's, of course, an especially tragic disease because people lose memory, and they lose many of the wonderful characteristics that make us uniquely human. But this is a slow process that takes many years to develop. And we can diagnose Alzheimer's disease relatively early in the course of the disease, providing an opportunity to intervene early to prevent or at least reduce the extent of ongoing cell loss. And what we are showing in these animal models of Alzheimer's disease is that the delivery of a growth factor by gene therapy can indeed prevent what would otherwise be the death of types of cells that degenerate in Alzheimer's disease. Now, let me say that these animals don't get Alzheimer's disease.
Right.
We mimic Alzheimer's disease by causing injury in the brain. But there's no other species of animal other than humans that actually gets Alzheimer's disease. So we can't test this in animals with Alzheimer's disease. We have to do the best we can, which is to induce the type of cell loss that occurs in Alzheimer's disease, recognizing, however, that we really won't know if this is effective until we test it in people with Alzheimer's disease.
And what results have you seen when you mimic the spinal cord injury?
Spinal cord injury addresses a different set of issues than Alzheimer's disease, as one might expect. In Alzheimer's disease, the main problem is that cells are degenerating and dying. In spinal cord injury, the problem isn't so much cell loss as it is a disconnection between cells. One can think of the spinal cord as a big telecommunications wire with about 1 million wires inside the spinal cord traveling back and forth, connecting different parts of the nervous system to one another, relaying signals to move a muscle or to perceive a touch or relay some other function like bowel and bladder or sexual function. And in spinal cord injury, the problem is not that the cells controlling those functions die, because actually very few cells die, but rather that the cells become disconnected and can't communicate with one another. And again, we're talking about a pretty substantial problem. About 1 million of these connections become disrupted. And our job there is to induce the new growth of these injured connections and to have them reconnect with their appropriate targets, because they, just as you can imagine with a telephone wire, you can't just connect up 2 random wires. They have to connect up to where they were supposed to go originally. And that's more or less the job confronting us. So what we try and do with gene therapy in the injured spinal cord is to implant cells into the spinal cord that secrete these growth factors that can produce the new growth of these injured connections. But in addition to stimulating the new growth of these connections, we also have to give them a scaffold to which the growing connections can attach and pull themselves along to find the appropriate target. So the gene therapy approach in spinal cord injury is sort of a twofold approach. One is to deliver the growth factor, but the other is to provide a scaffold upon which the regrowing connections can attach and extend.
Can you explain further how you do this?
Well, what we do is we take cells then from a rat in this case. We genetically modify them to make one of many different types of these growth factors. And then we implant those cells in fact, a collagen scaffold into the spinal cord injury site. In response to the growth factors secreted from these genetically modified cells, the injured connections from the spinal cord regrow, and they actually penetrate the injury site in large numbers. And we have found that with certain growth factors, they will in fact grow around the injury site and reestablish some connections downstream from the injury site and result in partial functional recovery. Before we can think of taking this to human patients, there are a few things that I believe one needs to do. One is that we need to show that these findings from rat models are really effective in larger animals, because the human nervous system, the human brain and spinal cord are quite different from the rat spinal cord. And it's important to show that these sort of approaches are practical in animals that at least somewhat more closely resemble the human. So that's one thing. Secondly, we have to show that one can obtain these results reproducibly, that it's the result of more than just one experiment. And so far, in fact, it is. A third step is to show that these experimental approaches are safe, that they don't cause more damage than good in the animals that they are subjected to. So these are sort of standard safety experiments that are done to make sure that everything is okay, and in fact so far those look pretty good. And so once one has all of that information, if things still look good, if one still has effectiveness in larger animal models, then one can begin to start thinking about taking these to humans.
With such promising results in ex vivo gene therapy, where would you like to see this research take you in the future?
Well, if we had enough funding to do additional studies, then what we would like to do, for example, in spinal cord injury experiments, is test rats with chronic spinal cord injuries. Most people with spinal cord injuries are in the chronic stages of their injuries, where they've had the injury many years before, and the real question is, can this promote recovery after chronic spinal cord injury? Those studies are very expensive, in fact, because it's hard to maintain animals for long periods of time under experimental conditions, and so those would be quite expensive but important studies. We'd also like to take some of this work to larger animal models, as I mentioned a few moments ago, to establish the validity of performing this kind of work eventually in humans, to try and find a useful clinical therapy for diseases that are largely untreatable today. And those studies, too, are quite expensive.
Thank you, Dr. Tushenskiy, for taking time with us today, and we wish you continued success in this very important work.
Thank you very much.
Thanks for watching. We look forward to you joining us next time as we take a look over the science horizon.